So thank you, Louise, and I'll also like to thank the organizers for inviting me to present a part of my thesis work on the CTD brain organoid model. So today I will focus on the results we have obtained regarding neuronal deficit and protein profile changes in this new human CTD model. So first I will introduce the context of the subject, which will allow me to present you the objective of my thesis and the experimental strategy. And then I will present the results, some results, and I will conclude on the main findings of my thesis and the perspectives of this study. So as you know, the CTD is a disease linked to a mutation on the SLC68 gene located on the X chromosome. And many mutations of different types have been identified. Some of them are represented here on the schematic view of the creatine transporter. And these mutations render the transporter absent or non-functional. However, for some mutations, such as the one with an asterisk, the creatine transporter has a residual activity. And some patients with these mutations may have less severe symptoms. This shows that there is a real clinical heterogeneity, which shows the importance of studying these different mutations in preclinical models, since they could potentially lead to different responses in treatment, depending on the patient and his particular symptoms. So treatment strategies currently being investigated aim to increase the brain creating pool, either by restoring a functional creatine transporter, by gene therapy or by refolding therapy, or by using prodrugs that can reach patient cells, brain cells, without the transporter, while still being able to perform creating function. Among these prodrugs, our lab has developed the dodecyl creatine ester, the DCE, which is a more lipophilic version of the creatine, which enabled it to cross membranes by diffusion. So restoring the brain creating pool is the main strategy being investigated, but we could also focus on testing molecules that will act on the consequences of the lack of creatine. And that could be used as a complement to the first strategy. But to do that, we need to better understand CTD pathophysiology. And however, nowadays, the preclinical models used to study CTD pathophysiology and to test drug candidates are quite limited. There are 2D cell cultures, which are almost exclusively made with patients' fibroblasts. So this is useful to study clinical heterogeneity, but this remains a very simple model, far removed from brain physiology. In contrast, there are CTD mice and rats, which are more complex than cell cultures, but they take little account of clinical heterogeneity. So there is therefore a lack of information due to the limitation of these models, a gap that in the context of other pathologies has tended to be filled for years by the development of 3D models, such as spheroids, organoids, or organ-on-chips. So this is a compromise between classical cultures and animal models, since they are more complex than 2D cultures, and they are closer to human physiology. And they can be obtained from human cells, especially patient cells. So to study the brain in particular, brain organoids are very interesting because this is a cluster of different cell types obtained from human pluripotent stem cells, and they can spontaneously develop some brain structures. They generally represent a fetal period of neurodevelopment, of human neurodevelopment, and they can be used to mimic a wide variety of neurological diseases. So the aim of my thesis was to identify the molecular and cellular mechanisms affected by creatine depletion through the development of a new human model mimicking the brain of CTD patients, namely brain organoids. So to do that, we had access to fibroblasts from CTD patients. We reprogrammed them into induced pluripotent stem cells, the iPS cells, and then we differentiate them into brain organoids. And after characterizing this new model, we identified the biomarkers and pathways linked to creating deficit in the CTD organoids. So initially, we had access to fibroblasts from 3 CTD patients with different mutations that are represented here on the slide. And in this fibroblast, this is the same type of mutation, that is to say, a deletion of one amino acid, but there is still a clinical heterogeneity because the patient didn't have the same symptom. In terms of writing, you'll see that the IPS obtained from these fibroblasts are marked CTD, followed by a number, which is the patient number, and a second number, which is the IPS clone amplified. So there is 1,4, 2,3, and 3,7 for patient 1, 2, and 3, respectively. So after validating the pluripotency state of these IPS, we checked the maintenance of the CTD phenotype in the CTD IPS cells. So with the creatine uptake experiments, we incubated the IPS with different concentration of creatine, and then we quantified the creatine inside the cells. So first, for the healthy IPS cells, we can see that there is an increase in creatine uptake depending on the initial concentration of creatine in the medium. And for the CTD IPS cells, we can see that the basal level of creatine in light blue is lower than for the healthy IPS cells. And there is no variation in creatine uptake at any concentration of creatine. So this shows that the creatine transporter was non-functional in these CTD IPS cells. To go further and validate our results, we also generated CTD rescue IPS cells by incorporating a vector containing a healthy LSC6A8 gene into the CTD IPS cells. And in the creatine uptake experiment, we can see that the basal creatine level of this CTD rescue cell is higher than for the original CTD cell line. And we can see that the creatine uptake is really high in these IPS cells, which shows that there is a functional creatine transporter in these CTD rescue IPS cells. So the uptake, as you can see, is really high, much more higher than for the healthy IPS cells. This could be explained because there are several copies of the transgenes that have been inserted into the genome of these cells. So this may be a limitation. But the basal creatine level, which correspond to our classical condition of cultures, tends to be similar to the healthy level detected in the healthy. So after, with all these IPS cells, we generated brain organoids using this protocol. So basically, we obtain embryoid bodies, which here are a cluster of different cells. That have aggregated by gravity thanks to the hanging drop method. And after several days of culture, we induce the formation of the neuroectoderm. This is this clear periphery here. And after, we embed the embryoid bodies in a matrigel bead, which is transparent here, that serve as a scaffold or support for the growth of neuroepithelial buds, like here at the periphery. And these buds subsequently differentiate during maturation to spontaneously reproduce some brain structures. So after 2 months of cultures, the brain organoids are fully developed. They can reach a few millimeters in diameter, and there is no difference in growth or final size between healthy and CTD organoids. We can detect 4 brain markers in these organoids. And the creatine uptake experiments also shows us that the basal creatine level in CTD organoids is almost 0 compared to healthy. And it is much higher in the CTD rescue organoids. For the creatine uptake itself, we can see a trend towards a creatine uptake for the healthy organoids, which is not significant here. But there is nothing at all for the CTD organoids. And there is a creatine uptake in the CTD rescue organoids. So therefore, we can say that we have generated for the first time CTD brain organoids without a functional creatine transporter. In these organoids, we can detect several type of cells, so neuroprogenitor markers with SOX2 and PAXis located in these structures that are ventricle-like structures. And at the periphery, we can detect several neuronal markers and also astrocytes markers and cyanotic markers. So overall, our organoids are composed of all the cell types and structures that are present with this type of model. However, for the organoids, the CTD organoids, we noticed that, as you can see here, the ventricle-like structures were less organized, smaller, or even absent. So this led us to look at the expression of these markers, the neuroprogenitor markers, by PCR, and we found that these markers were all underexpressed in CTD organoids, highlighting a potential neurogenesis deficit. And a previous study has already showed a deficit in adult neurogenesis in the hippocampus of a mouse CTD model. And we can also, so with our model, we can that model a more fetal period. We can also show a neurogenesis deficit at an earlier stage. And we can also, there are also some studies who show neurogenesis deficit in other brain organoid culture from modeling intellectual disabilities such as Down syndrome and Fragile X syndrome. So after we look at the expression of various synaptic markers, such as markers from long-term potentiation, but also glutamatergic and GABAergic markers, and all of these synaptic markers were also downregulated in the CTD organoids, confirming previous results of reduction of synaptic markers in the CTD mouse model. So with our new model, we have highlighted a potential synaptogenesis deficit as well as a neurogenesis deficit in the CTD. That could be the link between creatine deficiency and the impairment of cognitive function in CTD patients. So to understand the mechanical alterations, the signaling pathways altered and linked to this phenomenon, we compare the proteomes of our organoids using tandem mass spectrometry. So these techniques enabled us to identify thousands of proteins in the organoids. And we bioinformatically processed this data to narrow down the results to a few key proteins. So we did comparisons between organoids from a same patient versus the healthy cell line. And we identified 510 proteins, a set of 510 proteins, whose abundance is altered whatever the patient mutation. And there are also proteins whose abundance is altered, specifically altered depending on the mutation. So of all those proteins, we selected, with a bioinformatic analysis, 32 proteins, the top ones that are all connected, as you can see. And these proteins are linked to patient symptoms, but also to other intellectual disabilities, which further validate the relevance of our model. And we wanted to validate these results with new organoid productions and new conditions. So, for example, the CTD rescue experiment, not present here. So we selected the most significantly altered proteins, MAP1B, PAC1, and GSK3 beta for further analysis. So MAP1B is a microtubule-associated protein that influences the regulation of the structures of neurites, so potentially synaptogenesis. And PAC1 is a kinase involved in neurogenesis. So in our proteomic analysis, these proteins were overabundant in the CTD condition. So by Western blotting with new organoid production, we confirm this overabundance, and we can also see that there is a restoration of MAP1B and PAC1 level following creatine increase in the CTD rescue organoids. For the third protein, GSK3 beta, it is particularly interesting because it's a kinase involved in many neurodevelopmental processes, including neurogenesis, and it is active at the basal level, and it needs to be phosphorylated to be inhibited. So we checked the phosphorylation status of this protein, and hence its activation by Western blotting, and we, as you can see, we found a less phosphorylation of this protein in CTD organoids compared to the healthy, which means that this kinase is overactivated in the CTD condition. And when there is a restoration of creatine level in the CTD rescue organoids, there is also a restoration of GSK3 beta phosphorylation. So this kinase must be less active. So to conclude, we were able to show that the reduced creatine levels in CTD cells overactivate GSK3 beta that can act on several processes. So by acting on transcription factors, it can modulate gene expression, but it can also act on microtubule dynamics by acting on the protein associated, such as MAP1B. And this can maybe a beginning of a first explanation for the synaptogenesis and neurogenesis deficit and the impairment of cognitive function in CTD. And in a parallel study presented yesterday by Nicolas Tournier, we also show in the mouse model a deficit of glucose consumption with the FDG, which may represent a new CTD biomarker. And we try to understand this deficit with the intracellular mechanism. So we did with mouse and organoid model. And we highlighted that downregulation of a less activated AMPK, which can be also explained by GSK3 beta overactivation. And just to let you know, we have also identified a potential new CTD biomarkers linked to methylation processes with the DNA methylation, which could also influence CTD pathophysiology. And we have a lot of perspective for this study. One of them is to increase the diversity of mutations to be able to build a CTD brain organoid biobank, focusing on other type of mutations, such as missense, with residual creatine transporter activity, as well as using female cells to study the pathophysiology of heterozygous female patients. And we propose to use this CTD brain organoid biobank and the biomarkers identified to test the efficacy of our drug candidates, the DCE, and maybe other kind of supplementation to enhance some altered mechanisms. Finally, I would like to thank the association Xtraordinaire and the SCD for their support, as well as the patients included in the study and their families who donated the fibroblast. And I thank you for your attention.